Good afternoon, ladies and gentlemen. Welcome to this first Vice-Chancellor's lecture for 2011. It's a special privilege to welcome our guests, who I will introduce to you, our guest speakers, Dr. Francis Collins, Sir Mark Walport. Their CVs are many pages long and would take up all of our time, so forgive the very brief summary. Dr. Collins was at the forefront of the design and implementation of the Human Genome Project and was responsible for seeing it to its completion. He is known for his close attention to ethical and legal issues in genetics. He is a strong advocate for protecting the privacy of genetic information and has served as the national leader in efforts to prohibit gene-based insurance discrimination. Building on his own experiences as a young physician volunteer in a rural missionary hospital in Nigeria, Dr. Collins is also very interested in opening avenues for genome research to benefit the health of people living in developing and poor countries. And he shares this interest, of course, with our second speaker, Dr. Francis Collins.
And good afternoon to all of you. It's a real pleasure and a privilege to be here in Cape Town. But now let me turn to the Human Genome Project and how it fits with all this, because my charge here was to speak to you about genomics and how it plays a role in global health. And certainly genomics is something that I have a great deal of affection for, having spent some 17 years as the director of the National Human Genome Research Institute before becoming director of NIH. And let me say the Human Genome Project, I think, will stand as one of humankind's most ambitious enterprises, but it will also stand as a very fine example of an international effort, as Max Price said in the introduction, involving scientists actually in 6 countries from 20 laboratories, and with major support coming from our fine colleagues at the Wellcome Trust, the NIH, and others. But it was my job to serve as the field marshal of this enterprise, trying to organize what seemed to be initially an impossible effort to read out those 3 billion letters of the Human DNA Instruction Book. But happily, because of the very intense and creative efforts of thousands of scientists who saw this as a historic opportunity they wanted to be part of, and who decided early on that giving all the data away every 24 hours was the right thing to do, so that every scientist who had an idea about how to use it could get started right away. The Human Genome Project succeeded in that effort and produced a map and a sequence of human DNA, initially as a draft, then ultimately as a finished sequence, and now allows us to really begin to try to understand how to read this book. And it is a challenge because, of course, it's written in this funny language with 4 letters in its alphabet, but it's not a language that one can just look at and interpret. So a great deal of work now underway, and very exciting work, to understand the functions of the human genome. This has utterly transformed the way we do research. Graduate students today in human genetics can't imagine how anybody did this kind of research at all until you had the ability to just look up the sequence on the internet. In terms of its effects on clinical medicine, I think those are still modest, but they're coming. And they're coming in interesting ways for both rare and common diseases. They're coming for cancer, which I think Sir Mark may speak a little bit about. I want to say something, though, about what you want to know about the genome. Besides this initial sequence, which in the case of the public project came from a series of 5 or 6 anonymous individuals, and had no intention in that first pass to try to understand anything about phenotypic consequences or even to try to catalog variation in a systematic way. But as soon as we had that reference sequence, if you want to call it that, the next thing you want to have is the variations. You want to know, okay, we're not all the same. There's differences in our genome sequences, and some of those probably matter for medical reasons. Don't you want that catalog too? And that led then almost immediately, in fact before the genome sequence was quite done, to the formation of yet another international genome effort, the HapMap project, another project I had the privilege of leading, and which involved 6 countries, not quite the same 6, and a lot of support, again, from multiple sources, again involving the Wellcome Trust, with NIH very invested in trying to do this. But here, of course, if you want to look at variation, you'll have to think carefully in this initial pass where we could only afford to look at maybe about 300 samples. Where should those samples come from? The debates about that were intense. The decision was that those samples ought to have variation of some sort around the world, and I'll tell you in a moment what that looked like. But this did produce genotypes for 3.5 million variants, single nucleotide polymorphisms, SNPs, in 270 samples, giving you something about how common those variants were, the allele frequencies, something about haplotypes, that is, were adjacent variants traveling together or not. And this was then very useful as a means of setting up the opportunity to begin to scan the whole genome, looking for variants that were associated with disease risk, what we now call a genome-wide association study. And it also spurred a whole industry of developing highly informative, inexpensive methods of scanning up to 1 million of these SNPs in one experiment in order to be able to say, look at 2,000 people with diabetes and 2,000 people who didn't, and begin to see what the differences were. Now, where did the samples come from? Well, here's an example of somebody who's involved, who is actually carrying in his hand there the consent form for the HapMap project. This is a chief in Nigeria, because, in fact, we thought long and hard about how to do this and felt that it was important to get samples from Africa, as well as Asia, as well as individuals whose origins were from Europe. And we also felt it was important to do this in a fashion that engaged communities so that people had a sense of how these samples would be collected and described. Community advisory groups were involved in this. Charles Rodemy, again, was very much instrumental in setting up how to do this. And in this case, this community advisory group in Ibadan, representing the Yoruba, was a good example of a new kind of interaction between research and communities. The consent was both individual as well as this community consultation process. HapMap did not intend to try to connect variations with phenotypes, but simply to have anonymous samples where variation could be described, recognizing that the connection to phenotypes would be a later step, which I'll come to in a moment. But just the same, it seemed important that benefits to the participants ought to be provided. And so, in fact, that was the case, and this will certainly be something we will be talking about with the H3 Africa study. So the HapMap samples, 270 of them, you can see were divided in this fashion, with 90 from the Yoruba, 90 from individuals in Utah with European ancestry, 45 Japanese from Tokyo, 45 Han Chinese from Beijing. Not to say that anybody should think of these samples as being more than just that. Certainly you would not want to say Africa is well represented by 90 Yoruba from Nigeria. But as a start, this was actually a pretty useful database. It has gone on to become now a much more intense effort to survey variation in the genome by the founding and conduct of the 1,000 Genomes Project. And the 1,000 genomes actually has become 2,500 over the course of recognizing that more is better. And again, in this instance, this includes a search not only for single nucleotide polymorphisms, but for other kinds of variants that are more complicated, insertions, deletions, copy number variants, structural variants, and so on. And because so many samples are being sequenced, this should be a pretty comprehensive view of any variations in the human population that are present at a frequency of 1% or greater. And that means that if you're looking for a variant of that frequency that's associated with a common disease, it should be in the database. It's just up to you to apply this database to disease cohorts in order to find the ones that are going to be conveying that kind of risk. So these 2,500 samples have no identifying or medical data. The samples you can see listed here are from various parts of Africa. African Americans are included, Mexican American, Puerto Rican, Colombian, and Peruvian. Several different groups in Asia, as well as in South India. So this is, while again, nothing like what one would call a comprehensive sample of the human population, it is certainly more broad in its reach than HapMap was. And all of these samples, again, being collected with care as far as the consent process and the community consultation. So the first view of this, published last October in terms of a pilot, and preparing for phase 1 data release coming up on 1,004 samples. This will certainly be yet another of those sort of landmark efforts in understanding the human genome, along with the original sequence, along with the HapMap, and now the 1,000 genomes. But again, these provide you with a foundation of information. They don't tell you within themselves anything about actual phenotypic consequences. For that purpose, investigators have been using this data to try to identify variants that do predict risk for common diseases like diabetes and hypertension, like the common cancers or asthma, or multiple sclerosis or Crohn's disease. And the progress here has been breathtaking in terms of the numbers of discoveries. This is a cartoon showing you the human chromosomes 1 through 22 and the X and the Y. And each one of those colored circles represents a well-validated genetic variant that's associated with risk for a common disease. Each of the colors is a different disease. It would be difficult at this point to think of a common disease. It's not represented here because this has been applied so strongly over the course of the last 4 or 5 years, which has become possible because of HapMap and now because of 1,000 genomes. Now, each of these variants is here, for the most part, contributes rather modestly to disease risk, and so this has not turned out to be incredibly valuable as far as personalized preventive medicine to predict individuals' future risk for disease, although there is useful information here. But what this has done is to point towards pathways that are involved in diseases that we didn't know about before, and that create new ideas about therapeutics that are actually quite exciting in many instances. So this is sort of the first wave of the harvesting of what we can learn using this foundational information about human variation to discover what are the molecular contributors to common diseases, the things that in general have been so vexing for us to understand. My own lab works on type 2 diabetes, adult onset diabetes. And we are now up to almost 60 variants that have been discovered that play a role in risks for that. And they make some sense in terms of pointing to certain pathways, but those were not pathways that we knew about for the most part. It also turns out that of those 60, we've discovered most of the known drug targets for type 2 diabetes. Of course, we knew about them already, but it's interesting, they pop up in this screen. And that tells me that there are probably some others in that list of 60 that we didn't know about that would also be very important as new drug targets. And this could be, in fact, one of the most significant developments in terms of identifying new therapeutic ideas instead of continuing down the same paths that we already have known about. This approach has played out in various interesting ways in various populations, and I just picked a few examples from these genome-wide association studies, and maybe particularly ones that I thought might be of interest and relevance in Africa. Here's a publication from Charles's group, the first GWAS study in African ancestry populations, in this case on hypertension and blood pressure, and coming up with hits on a number of interesting genes, including a sodium-potassium-calcium exchanger, which makes a certain amount of sense, a voltage-dependent calcium channel, and others. And again, also on that list were genes that nobody would have predicted could possibly play a role in hypertension from what we knew about it then. But again, I hope you get the sense here of the power of this approach because it is comprehensive. You don't have to have the right hunch. The genome is a bounded set of information. And if you have a method that can scan the whole thing, you're going to come up with answers that don't require you to have the right guess when you start. And that kind of approach, where you can really expect to be able to get the entire set of answers instead of just looking under the lamppost to find your keys, which is what we've often done in the past, is truly exciting. Here's another one about infectious diseases, because clearly individuals have different susceptibilities when exposed to a pathogen. We know that's true for HIV. We certainly know that's true for malaria, the long-held understood story about sickle cell carriers being relatively immune, for instance, to cerebral malaria. But we also know this now for things like tuberculosis, because here is this paper from Nature Genetics last fall doing a GWAS study and identifying a susceptibility locus for TB on chromosome 18, and there are other such findings that have been reported in other parts of the genome. And again, if you can identify variants in the genome that confer either heightened or reduced risk for a disease like tuberculosis, that shines a light on the pathogenic pathway that the pathogen uses to cause disease, and that might turn out to be very useful therapeutically. One that I really like here in terms of therapeutic opportunities is about sickle cell disease. So sickle cell, after all, is a disease which in most patients causes severe difficulties beginning by about the first couple of years of life, with the sickling of the cells leading to obstruction of small vessels, to painful crises, to damage to organs, particularly to lungs, heart, kidneys. And yet, we have also observed over the course of time that some sickle cell patients have a milder course. And when you look to see why that is, a major reason is that some people actually have higher levels of fetal hemoglobin, and that seems to be protective. Now, fetal hemoglobin is that hemoglobin that you make primarily in utero, and then it gradually turns off, beginning a little bit before birth and completing within the first few months of life, and the adult hemoglobin, which is the one with the sickle mutation, comes on. But some of us don't turn that hemoglobin F quite all the way down, and if you happen to have sickle cell disease and a higher hemoglobin F, it's going to help you. So what is that about? What is it that allows fetal hemoglobin to stay on? This is a good question that you'd like to know the answer to, and this is very susceptible to this genome-wide association study. So what was done was to, in fact, show in a GWAS study that there's a very strong signal for a gene called BCL11A that plays a role in fetal hemoglobin. So BCL11A, there's two alleles here, as I hope you can maybe see in the lower right, but it's a little faint here. That actually has a very significant effect on what level of fetal hemoglobin you make as an adult. And it also turns out that has a very significant effect if you have sickle cell disease on what level of hemoglobin you make, hemoglobin F, and it also means it has a significant effect on whether you're going to have severe or mild disease. It turns out that BCL11A is a transcription factor, and it turns out that the version that leads to shutting down hemoglobin F is the one where you have higher levels of a particular isoform of this. So it normally suppresses hemoglobin F. So if you're thinking about making a drug, that's just the kind of thing you'd like to have, because it's always easier to suppress something than to activate it. So the idea here is make a small molecule, suppress BCL11A, increase hemoglobin F, and that might help sickle cell disease. Now that is a hypothesis that hasn't yet been proven, but a lot of progress has been made in just a couple of years. And I think this is one of the most exciting developments in the potential for new treatments for sickle cell disease in a very long time. Here's one that I think also is particularly interesting from what it tells us about the interplay between selection on the human genome, which has been going on and continues to go on, and how that plays out in terms of other consequences. And this is the discovery of a variant in a gene called ApoL1, which turns out to have two different effects. It turns out that the variant protects against sleeping sickness. And so you could imagine in parts of Africa where that is endemic, that having the protective allele would be good for you and would result in increased survival. But what we also have discovered is that this same variant may well account for the fact that Africans and African Americans have a significantly higher instance, 4- to 5-fold higher instance of a kidney disease called focal glomerulosclerosis, which is relatively uncommon in those with European ancestry and much more common in those with a background from Africa. So it does seem that these are actually two sides of the same coin. That in an area where sleeping sickness was common, the protective allele was selected for, but actually that same allele can cause you trouble and result in this kidney disease. So it's a play, a back and forth between benefits and risks, and it is certainly an example where selection has left its mark on the genome. Perhaps the most famous of those, of course, is the sickle mutation that I talked about a minute ago, but here's another one, and there are many more. Pardis Sabeti has been looking across the genome for evidence of recent selection and found hundreds of examples where that seems to have happened in various parts of the world. Many of them, it seems, are going to be explained on the basis of infections, which have brought particular alleles to higher prevalence because of their protective ability. But not all of those will be benign in other circumstances, as this example shows. I'm not going to say a lot about cancer, but I suspect Sir Mark may very well touch on this, because I do think this is one of the main exciting payoffs right now of the ability to survey the genome, because after all, cancer is a disease of the genome. It comes about because of mistakes in DNA, either inherited or acquired during life. And again, because of the ability to use genomics in this sort of way, we can begin to ask and answer questions that previously puzzled us, but we weren't quite sure how to approach them. For instance, Funmi Olopati is trying to understand, why do African American women develop more aggressive breast tumors than women with European background? It's clear from epidemiology that's the case, but is that because of poor access to health care, or some environmental influence, or some genetic influence? Funmi is studying breast cancer genomics in West Africa to try to understand, are breast tumors in some way systematically different as a group in this situation? That doesn't prove to you whether it's genetic or environmental, but it would tell you something more about where to go and look next. And these kinds of approaches are now possible and would not have been a few years ago. I want to finish, though, by saying that I think we have some real challenges ahead, and I'm delighted to be here for the H3 Africa meeting beginning tomorrow, because one of those challenges is clearly that we need to focus research efforts where the opportunities are, and they don't all happen in the countries with high incomes. And we also need to therefore work on building capacity in countries around the world to do this research, because it is exciting. It is a great moment scientifically. And I'm fond of showing images from this website, which, if you haven't discovered it, I would encourage you to go and have a look, called World Mapper. Because what they do with this website is to take the map of the world, which is here on the familiar basis based on landmass, and then they look at various parameters and say, okay, what would the world look like if you factored that in? So here's landmass. Now what happens if you, for instance, pick a disease like HIV/AIDS and ask, where is it most predominant? Look and see what happens. So you can see the way in which Africa has occupying now almost most of the center of the picture. India also coming up very big. Lots of the rest of the world almost now shrunk back to little squiggles, except maybe for Haiti, which you see here. Dramatic ways to show evidence, which in tables is impressive, but somehow this seems to hit you harder. Non-communicable diseases, interestingly enough, while we think of those, at least I think many people do, as primarily diseases of high-income countries, and certainly there's plenty of that going on here in the US and in Europe and in Japan. Look at China, look at India. Africa here not coming up as fast yet, but if you had a trajectory, this would change over the course of time. Here's South Africa down here. Now what about capacity for doing research? How would you look at that? Well, one way is to look at publications. So what does the world look like if you look at scientific publications? You see the problem here? We've got to fix this. Because look at this sort of bloated United States over here, and look at all of Europe, and Japan, my gosh, is like swallowing up half of the right side of the picture. This is South Korea, this big green blob. And of course, where you look and hope to see much more opportunity in the future, here is Africa, and that's South Africa, that pink part down there. This is certainly something that could become an opportunity for great change. And I think those of us who are interested in global health are determined to make sure that that is one of the primary goals of everything we do. Capacity building is going to have to be done as true partnerships. It's going to need to involve investment in institutions as well as individuals. It's going to require replicating success stories, and it is going to require looking at what has worked and what has not. So I'll just finish with one more quote, and this is from my favorite South African, who happens to be a friend, Archbishop Desmond Tutu. You probably know he had his genome sequenced as part of the effort to undergird our shared humanity. And he quotes here in a piece that came out of Science magazine just a couple of weeks ago, "My hope is that my genetic code may provide a voice for the region and serve as the starting point for a map of DNA variation significant for Southern African peoples to be used for medical research efforts and effective design of medicines. My dream," says the Archbishop, "is that by including all peoples in understanding and reading the genetic code, we will realize that all of us belong in one global family, that we are all brothers and sisters." Wow. And I echo his wow. What an amazing time this is. If you're new to science, perhaps it hasn't sunk in yet what an amazing transition we have made in the last decade. From poking around areas that we understood poorly to having comprehensive abilities to ask questions. And there could never be a better time than to take that opportunity and spread it to investigators across the world. And so it is with great pleasure that I have a chance to be here with all of you here in Cape Town to take part in what I hope will be a very significant few days where we bring together the kind of talent and creativity that it's going to take to make the most of this opportunity, to understand what are the causes of illnesses that have puzzled us for all time, and to figure out what we might do to prevent and treat them. We should have no less ambitious goals than those. Thank you very much.